Okay, so I think we're going to get started. Most of the people from the waiting room are able to join. So hello, everyone. It is my pleasure to launch this science visualization workshop series and welcome you to this first session. Today's topic is Introduction to Scientific Visualization and Drawing Techniques. My name is Joanna Konopka, and I'm a postdoctoral fellow in the Department of Neuroscience at Johns Hopkins University. Daria Task is a postdoctoral fellow also at Johns Hopkins University in the Department of Biology. And together we are your organizers and moderators for this workshop series. Our partners for this workshop series include Project Bridge and the Doctoral Life Design Studio at Johns Hopkins University. And we want to give special thanks to Andrea Pereira from Doctoral Life Design Studio, who has been tremendously helpful in setting this up. Our generous sponsors are the Kavli Neuroscience Discovery Institute and the Department of Neuroscience at Johns Hopkins University. We would like to acknowledge and thank all our partners and sponsors for making this event possible. All right, so before we move on to the actual workshop content, we would like to share some information about the workshop registrants. As of Monday morning today at 10 a.m., we had 1,574 registrants from a total of 85 countries. Most of the registrants are from United States, India, Germany, Great Britain, and Canada. And in terms of registrants' roles, we have registrants at all career stages, including more than 50% of our registrants being trainees. We also have a wide range of experience levels, as well as registrants that are interested in all sorts of art media. Whichever part of the world you're joining us from, and regardless of what is your career stage or your experience level, rest assured you're not alone. There are other people who are just in the same situation as you. And we're very happy to have all of you join us today, and we look forward to your unique perspectives and contributions in this workshop series. So I will now pass this on to our speaker, who is not only a scientist, but also a very accomplished artist and an illustrator. Please welcome Dr. Barrett Klein, who has prepared some amazing content for you today.
Huge thank yous to Joanna and to Daria and the fine folks at Johns Hopkins. And thank you all for me in the ether today. We have 3 hours and I have an ambitious program. We'll see if my timeline pans out at all. But I'd like for us to practice with both analog and digital means of creating scientific illustrations. And there are 101, 1,001, 1,000,001 different ways to approach visualizing science. I want to talk about some general guidelines by which we can achieve some graphical excellence. And I'll talk about what that means. Let me share my screen. But before I do, I want to introduce not just this workshop, but the whole series. So as many of you know, this is a series of 6 scientific visualization workshops, and I'm bringing along 3 expert friends for half of them. So this first one, I'm going to go through some drawing techniques, and I'll think with you about how to do that basically with pencil on paper, but also some digital means for drawing and manipulation of media. And we'll talk more about that as we go along. The second workshop I called How to Avoid Lying When Using Visuals. And this will be an opportunity for everyone to submit works of theirs, visuals of all kinds, whether it be a poster, maybe a figure that you're submitting for a peer-reviewed publication. It can be all over the map. It could be a logo, a billboard image. And this kind of content will collectively critique and improve. The third one will be run by a former student of mine who's become an amazing professional medical animator, Danielle Van Brabant. The fourth workshop will be Reaching a Broad Audience, and that will be with my friend Damon Kylo, who lives in Cuba, but will join me for a day in La Crosse, Wisconsin, and that's where I'm coming from. I'm in my home right now, and I work at the University of Wisconsin La Crosse along the Mississippi River in the driftless zone, United States. The fifth will involve designing logos, and I'd like for all of these workshops to be participatory, so I'd like for you to produce your own logo, and I'll guide you along with Amy Zhang, who's my co-lead on that. The last one is extra special visualization challenges, if you're up to the task. I love questions. I love tangents. I won't be actively reading the chat, but Daria and Joanna said that they would keep tabs on that. If there are relevant questions that you want to ask at the moment, maybe we can interrupt for at various points, but we'll definitely have tea breaks, bathroom breaks, cookie breaks. My mom wants to eat a cookie with her tea, and I know she's out there in the ether with us. And during that time, I can also answer questions. Okay, I'm going to share my screen, and I'm going to start with an introduction of me, so you know why you're bothering to join me as your lead in this endeavor. Let's see. All right. So that's me, Barrett at pupating.org. I lead the pupating lab at the University of Wisconsin-La Crosse. And I'll start by introducing, my science. So I'm a biologist. I'm an entomologist and an animal behavior biologist, and I like to look at social insect behavior. So here I took thermal imagery of a honeybee colony, and they're presently mobbing an invading wasp. They're baking her alive. They're suffocating her with an envelope of CO2, and the thermal imaging allows me to see differential temperatures emitted by those bees and the baked wasp. I look at sleep biology, sleep in honeybees especially, and here I've glued a little piece of ferrous or magnetic metal on the back of a honeybee, and I study her behaviorally. Sometimes there are consequences with that little ferrous metal. I wiggled her with an insominator, a bunch of magnets for one night, and the consequences of sleep loss for these bees played out in their communication. You see this bee, for example, this bee had that ferrous metal tag, and when she engaged in a waggle dance where she communicates direction and distance information to an advertised site, it's a little bit sloppier. The directional component is variable versus the one, a copper bee, that wasn't manipulated in the same way. So a follower of this bee, this less precise dancer, will switch dances. Go to maybe, hopefully, a more precise dance. And then the one that gives the more precise dance, her follower will more likely exit the nest. And sometimes I'm able to look inside the bee of a brain and see how they're able to process odors during sleep. These 4 quadrants give you an idea of how some of the biology that's conducted in the pupating lab really thrives on visuals. So that's by day. By night, I'm a vigilante, righting the wrongs of the world by wearing apparel that's made of insect products. Sometimes I'll put recycled bicycle helmets in a hive, and the honeybees will build their comb on top of that. So I found, after I did my undergrad and before I did my graduate studies in entomology and behavior, that I could pursue the intersection of science and art in really interesting ways. So I made natural history exhibits. So under that giant marbled orb weaver spider that I helped build, I was working at a Chase studio, a natural history display making studio in the Ozarks. And then I moved on to the American Museum of Natural History in New York City to continue making natural history exhibitry. And that included making giant viruses, platyhelminths, and bacteria and other organisms for outreach, for education. And that meant communicating with scientists and being that bridge, that conduit between the science and the design. So for example, here, those viruses are 90 nanometers across, so they don't even reflect visible light to the human eye. So we made up the colors, and the color coding helped viewers distinguish the different viruses. And I found that using these tools of an artist, whether it be direct fabrication, molding, casting, I could work on science in novel ways. So I teamed up with Ryan Taylor at Salisbury University and Mike Ryan at UT Austin to make faux frogs, robotic frogs, with an animatronics expert, Joey Stein, in Brooklyn. And so these methods I would use in making exhibits. And here we were making frogs that could call to females. So you have these ridiculous little male robots that are calling to females, and the female will hop over, surprised, shocked, dismayed, in disbelief that the male is not responding to her advances. But this allowed us as a team to ask questions that you could otherwise not ask of a real frog. In multimodal signaling, how important are these aspects of visual signals versus these acoustic aspects of signals? Occasionally, I'll try to convey concepts or subjects in science for others. So my friend Kirsten Robling in Germany first described a new subfamily of ant, and that's to the left. To the right is a gobbeting honeybee, and that appears in Piping Hot Bees and Boisterous Buzz Runners, Tom Seeley's latest book on honeybees. And if we have time today, I'll walk you step by step how I created these two, as well as this third one, which is the most recent. And this looks at Danielle Russler's really powerful analysis of jumping spiders. So you see this jumping spider dangling in front of the moon on a silken thread. This jumping spider is asleep. And not only is she asleep, but you can see I highlighted them in red, these retinal tubes. And the retinae dart about during rhythmic, frequent, recurrent stages of this sleep, looking a lot like rapid eye movement sleep in, say, this mammal or this bird and this other reptile, or maybe this octopus. So in this phylogenetic tree, you're looking at a very distant relative that may engage in this behavior. Again, if we have time, I'll go through how I produce these and what layers of meaning are embedded in these images. Lately, I've been really excited about writing books. So next month, my first book, The Insect Epiphany, with a gorgeous cover by Elizabeth Jean Younts and visuals throughout by different artists and others, looks at how insects affect human culture and have throughout history and across the world. So how our six-legged allies shape human culture. And in 2 weeks, my colleagues, Niels Rattenborg, John Leskew, and I will turn in our first draft for a book about sleep across the animal kingdom. And my mother, Karen Ann Klein, has produced the visuals throughout. This one is a tree of sleep, a phylogenetic tree where the different lineages who have been studied with regard to sleep appear across the branches. And there are messages in that as well. So now I'd like to dive away from who I am and what kinds of things I've worked on and think about graphical excellence, a little bit about the history of visualizing science, and some powerful imagery, and why we'd even bother visualizing science. Rewind 100,000 years ago, and here's an ancient, very early, maybe, artist studio. A bivalve or clamshell with some ochre pigment and some tools. Now fast forward 100,000 years and you've got a billion pixel array of the Milky Way galaxy. And if we think about what's happened in between those two, we've got I could share thousands of different fabulous images that have changed the way we view our place in the world, in the universe, and certainly science. So for example, in the center here, you see the heliocentric or sun-centered solar system, where Copernicus challenged Catholic dogma to say the Earth isn't the center of everything. And then to the left, early microscopist Robert Hooke produced an image of a flea during the time of the Black Plague. To the right, Leonardo da Vinci looked at embryonic development as well as lunar properties, water properties in his famous notebooks. On the lower left, most famous image of X-ray crystallography by Rosalind Franklin, where Watson and Crick from this image could very carefully derive not only the helical structure of the molecule of the century, deoxyribonucleic acid, but also measurements within that molecule. And then you've got Ernst Haeckel's glorious images, in this case of Cnidaria, but across animals and beyond. And then Vesalius, departing from pigs, looked at humans for medical tutelage. And finally, you've got Röntgen, who got the Nobel Prize for discovering X-rays, and this is his wife's hand with her wedding ring. The list goes on, but I'll point to the most famous doodle in all of biology, and this is in Charles Darwin's notebook, which has since been stolen, I've read, in which you've got, I think, and then the first visual metaphor of evolution in the form of a tree. How do you read this tree? Well, whatever C is, maybe it's a finch, and whatever B is, and D, if you travel along their branches, they share a most recent common ancestor here. Go deeper back in time into this node, and you share a common ancestor with A. So all of life on this tree shares a common ancestor back here. This was such a powerful visual. Ernst Haeckel went crazy with it. And in 1859, on The Origin of Species, Charles Darwin made this on the right the only visual in the whole book. It's a fold out showing possible multiple origins of life in these trees, with these stunted branches representing lineages that went extinct. Well, all evidence suggests that we have a single origin of life, but he was positing a hypothesis of multiple origins. So as scientists, how can we visualize our data? One way is through alphanumeric tables, columns, rows. That's pretty abstract. It takes some work, but sometimes it's very important, even essential to include in our science. Or we could have charts. We could have life cycles, nutrient flow charts. Those can be helpful. They're also pretty abstract. We can have 2D plots or their ilk, XY plots, for example. Or we could have what I argue is the least abstract, an image, whether it be a photograph, a video, or a two-dimensional illustration. And that'll be our primary focus today, although we'll look at some of the other image visualization devices in other workshops. Now, visualization can take 3D form, and some historically exquisite forms were used for educational purposes, and a father-son team of the Blashkas, blue glass, and that mollusk on top and that cactus below, all 100% glass. And to the right are some of the famous La Specula wax anatomical models that were used to teach medical students biology. Case in some, you could even dissect the woman lying on her satin sheets to look at internal morphology. Here's a thermal animation I took of a bee inserted inside a honeycomb cell. Here is her head, here's her midsection, here's her hind section. You see that that hind section is continuously ventilating or respiring. And the midsection is piping hot, if you look at that thermometer there. Well, I wouldn't know that she's hot unless I were using a thermal imaging device, but I can look at the continuous ventilation and make correlations and say, okay, this bee inserted in a cell is not sleeping. She's actually a heater bee, heating developing bees in adjacent co-cells. Tom Eisner with Doc Edgerton at MIT took this super high-speed imagery with 16mm celluloid film to show a bombardier beetle shooting like a Gatling gun out of the tip of the abdomen boiling liquid. And this appeared on a US postal stamp. Here is a still image. And by using that high-speed imagery, Tom Eisner was able to say more precisely how a bombardier beetle is able to expel this liquid when no other organism can without burning up the butt. This, as well as previous examples, I would say are beautiful examples of the power of scientific visualization in terms of mining data, finding specific data, or analyzing data. You can do this with 2D plots and points laid out. Maybe there's a clumping of data. And here, potentially novel discovery, new discovery, which can lead to further testing and further hypothesis generation. So a lot of value to visuals embedded in your science or driving your science. My twin brother, Arno Klein, would produce images of brains for medical use. And here are just a number of ways you can take a brain. You can inflate it, you can color the anatomical features, functionality, all manner of ways to better understand that 3-pound wonder in our cranium. So medical use of imagery is important, from protein folding to the surface structure of viruses. Whether you're a biologist, a psychiatrist using a Rorschach test, a chemist, a physicist, imagery can be key to conveying your science accurately, clearly, efficiently. And I'm going to turn to a giant in visualizing information. And this is pulled from Edward Tufte's The Visual Display of Quantitative Information, where he gives a list that defines graphical excellence. I'm going to skip to this one down here. I love this. Graphical excellence is that which gives to the viewer the greatest number of ideas in the shortest time with the least ink in the smallest space. Now, we can think about that, and we'll certainly think about that when we're with Amy Zhang talking about logos, because all of that is key. Down here, you see graphical excellence is nearly always multivariate, and graphical excellence requires telling the truth about the data. We always want to tell the truth. Now, there is some artistic license, and we can discuss when you deviate from the truth, in quotes, effectively. Graphical excellence, nearly always multivariate. What does that mean? Well, this image, let's look at it blown up, is Tufte's favorite. He always goes back to this one. And here you've got Napoleon's army marching to Moscow. And there are 6 layers of information here. For example, you've got not only where they went geographically, but the timeline that took place. You've got the thickness representing the number of soldiers that advanced and the piddling number that retreated, as well as other data, all packed by Charles Minard in 1869, a statistician, in this exquisitely efficient graphic. Now, a lot of people have copied this or used this in different ways, and whether they know it or not, they've used some of the techniques of Minard. For example, Maggie Nelson interpreted Tom Seeley's data for Honeybee Democracy in terms of, well, here's your hive, and the bees are advertising to a new home. And at first, actually, this is a swarm. They're outside of the hive, and they need to find a new home fast because they're exposed to the elements. So what happens? Well, one bee's advertising a site in this direction, this far away, and another this and this and this and this. Well, over time in the day and across the 2 days, you get this uniform 100% advertisement to a single site. And that's when the swarm lifts, swirls around, and heads unanimously to this one site. When we think about visualizing nature, we can see how images have impact, specifically in environmental awareness. So here's the famous Keeling curve that shows us how carbon dioxide has risen. Yes, it fluctuates up and down, but the big picture, radical rising to our detriment. In this case, you've got boats that are trawling for shrimp. And I turned away from consuming shrimp a long time ago because I see the devastation that's happening across the seabed here. And this individual said, imagining is one thing, but imaging is something else. When we see an image, it really crystallizes the impacts and an attitude, in this case, towards the sea. Cornelia Hesse-Hoeniger is an environmental activist, an artist. She was a scientific illustrator. She's co-published as a scientist. And here, outside of a nuclear power plant that faced a meltdown, it can be one of the cleanest nuclear power plants, say in Switzerland, but Cornelia Hesse-Hoeniger is picking stink bugs, pentatomid bugs, and finding that they're bilaterally asymmetrical mutants, she claims, from radiation. This was an article a dozen years ago about Steve Jobs. And in it, the journalist said, both Einstein and Mr. Jobs were very visual thinkers. The road to relativity began when the teenage Einstein kept trying to picture what it would be like to ride alongside a light beam. And then it goes on to say that Mr. Jobs would think with his designer, actually fingering foam models of the products they were developing. And models, two-dimensional, three-dimensional, are used all the time to better understand things. That's what Crick and Watson used. They resorted to building physical models to see what fit together perfectly to visualize deoxyribonucleic acid. So I want to pause for a moment and make you think about this question. When is an illustration useful to visualize a subject? Think about it. You have the option of just writing a bunch of text or creating a visual. Take a moment, maybe scribble down a couple of ideas, and I'll share some of my ideas with you.
Where would you like people to put their answers in the chat or just unmute
Oh,
them?
that'd be fun.
We've got simplify complex. Oh, they're coming in fast. Simplify complex systems in order to
Great.
more simple and intuitive solution to a problem. The scale of the subject is too small or too large.
Great. Actually, let's focus on a couple of those. The idea of something being very complex, making it simple. One of the challenges as a visualizer of science is going simple so that we can understand it better, but not at the expense of losing essential details or information. So how simple do you go? This is an ongoing challenge. What was another one?
About the scale? Is that what you just asked?
Yeah, okay, scale. If we go way small, think powers of 10. If we go super small, subatomic, or if we go super large across the universe or multiverse, how do we visualize that in order to better understand what that would even mean? Okay, so let me go through a few examples I have. First, I'll start with this video that I took and we modified so you could watch that follower. Oh, can you see there we go. Here's a dancing bee, but I'm highlighting this follower of that dancer. That follower, what is she doing? Has that bee faced sleep restriction or sleep loss? How will that follower respond? And that's a lot to look at. That's a lot to process, a bustling beehive. So instead, what we want to do is, yes, simplify. And you'll see on the bottom, there's a 2D plot, an XY plot. And we see that our independent variable is precision or imprecision of dance, namely the directional component of the waggle dance. And then the response or the predicted probability of exiting is our Y. And you'll see that over here, as I was talking about before, this is a sloppy dancer. So what happens? A honeybee following her will switch dances. While a more precise dancer, her follower is more likely to exit the nest. That's exactly what this says. But my thought is that this graphic may be faster to process. than the other graphic. I did tie them together because I found them both to be very useful in the publication. All right, so our first, when subjects are inaccessible. Albrecht Dürer produced that etching of a rhinoceros. He'd never seen a rhinoceros. This is from secondhand descriptions. And that one on the upper right, is it a unicorn? It's a narwhal. And you better believe that person probably never saw a narwhal. And in an article written about visualization, the journalist said, artists are still painting things they cannot see in real life. Rather than being separated from their subjects by thousands of miles, though, today's artists are separated by thousands of years, even millions of them. Now, these are subjects that are inaccessible because they're fossils. So we don't know what the real one looks like. When I was asked to draw this Hylonomus lizard, I was given a pretty shabby photograph of a lizard, of Hylonomus, of a fossil of a lizard. And then I thought about who are the extant closest relatives, the presently living closest relatives. And so I made up this image. Now, I didn't make it in color. The colors would have been made up. I did make up those slight contrast patterns on the back. But it's a hypothesis, a working hypothesis. How about when objects are hidden from view? Kind of related to the previous one. Digital morphology at University of Texas, Austin, where I did my PhD, uses high intense X-rays on a lot of different, like, for example, fossils. And they're able to determine whether the fossils are frauds or not, or visualize embedded fossils that otherwise you couldn't reach. And Camargo in Brazil produced beautiful images of subterranean stingless bee nests. Walker et al. Published this 10 years ago, and it's an exquisite view of the midsection of a blowfly and how the wings move, the bases of the wings, and the muscles that are powering those wings. I wouldn't be able to see this. Even in a dissection, it'd be a messy scene. So this visual really helps me see which muscles are pulling or contracting against which sclerites or armored elements. When salient features need to be emphasized, David Rubik, a stingless bee expert, wanted me to draw these long-tongued, and yes, this is a tongue of a bee, long-tongued orchid bees. And these males, if you look at them closely, have tufts of hair-like structures, and species to species, they vary. So if you want to distinguish one species of male orchid bee from another, these bees' knees could help you. When phenomena are infrequent or short-lived, ephemeral. So David Rubik also asked me to depict an orchid bee being eaten by an assassin fly, mimicking a different genus of orchid bee. Camargo again. This is, oh, by the way, this is rarely seen, but he knows it happens from evidence. And then here's another example where a bee has attached pollinia from a flower. Well, this is hard to, unless you have a camera going at all times, it's hard to catch that. Next, simultaneous display of complex information. Here's an elephant dung mound, and underneath this, you've got soap opera tales of what happens in the species of dung beetle, where the short-horned male is always defeated by a longhorn male. So he has other ways of getting at the female, and I won't go into each story, but this visual shows you how complex the different scenarios that can maintain the genetics of short-horned and long-horned male dung beetles can be. Here's a scene in Central Park, New York City, with little vignettes of the activity that goes on. And my mom and I did this illustration for a book on evolution in which we wanted to show, yeah, I think somebody's able to draw on my screen. How about that? You can add to it. So here we've got mouth parts for different orders of insects, and you see they're color-coded. So for example, mandibles are green, and this moth does not have mandibles at all. So while insects have these proboscis, in a lepidopteran versus a hemipteran, a moth versus a true bug, the origins of the components are very different. I'm going to stop there with a presentation on visuals in science and graphical excellence. And what I want to do now, what I'd like to do now, and I think, wow, we're 8 minutes ahead of schedule, which is just what I was hoping for. What I'd like to do now is take a 5-minute or so tea break. And while you're taking your tea break, the next portion will be an activity that I guide you through. So what I'd like for each of you to do is to grab a pencil, a pen, and an eraser, a ruler, and preferably something that's clear, but any kind of ruler. I'm going to use these calipers, but any kind of ruler, and a mirror or another human being. So in the meantime, take your tea break, grab those few materials, come back and join me, and we'll dive into the next portion where the view will be Barrett's hands rather than Barrett's mug. Also, if you want to, now's a good time to type in the chat questions, and maybe I'll spend a few minutes addressing some questions before we dive into the analog portion of creating a scientific illustration. All right, go to it. And Daria, give me hands.
Yes, sir.
Thank you. Do you want to take a pop poll?
Absolutely.
Okay. The question is, do they want me to draw a cicada, a bumblebee, or a Madagascar hissing cockroach?
Okay, we can do that.
Maybe I could address a question or two at this point about graphical excellence, scientific visualization, before we move on to one way of approaching scientific illustration.
There have been a few questions about your personal background or journey.
Oh, sure.
What inspired you to get into art and illustration? What were some of the other questions?
I grew up.
How do you choose between photos and drawings? What's best?
So right now I'm teaching a course on scientific visualization, and sometimes when I teach that, I have a session on macro photography where the participants take images of something small, photographs, and then they illustrate the same subject. They put them side by side, and by putting them side by side, we make comparisons about the value of having one or the other, or in some cases, both, because there's value in having both. Not only the authenticity that a photograph can often offer, but also the salient features that are drawn out from a visual of that, from that subject that was photographed. So my background, I'm raised in a family of artists. So my mom and dad, my mom is, as you know from seeing her gorgeous work in the slideshow, is the greatest living artist. And my father and mom started the Arnold Klein Gallery and ran that for 40 years in Michigan. My brother is a scientist and everything else under the sun, and visuals are at the core of almost everything he does. My sister is a luthier, and she works with her hands to produce visually exquisite musical instruments. And art was something that wasn't daunting for me, but actually really stimulating, inspiring, and exciting, and equally as important, accessible to me. And the science, I mean, from 5, falling in love with insects, I just wanted to try to combine the two in interesting ways. So for a lot of people, I understand that science can be daunting or art can be daunting. And I like to think that scientific visualization can facilitate the breakdown of these walls that would otherwise prevent you from engaging in one, the other, or both.
Do you have time for another one? There's an interesting question here, Gab.
Sure.
Also, Bumblebee won, by the way. But the question is, Do you ever feel constrained by having to illustrate biological accuracy rather than being able to express artistic innovation?
Well, there's room for both. So when you embrace your personal aesthetic, you can do so outside of science, like with those crazy honeybee helmets, right? You can do your art on the side that's inspired by science, that's infused with science, right? As well as bringing the aesthetics to your science. And I've seen a lot of really creative ways, both in interpreting, for example, origami folding of a ladybird beetle's wings through origami. So there are a lot of ways that you can use maybe less conventional artistic approaches to augment or clarify your science. So no, I don't feel hindered because I can play around with it in infinite number of ways. Now, it may be a context-dependent constraint that I feel like, well, can't do that here, but there are other ways. Sometimes when you're limited, you can become resourceful. But I'm happy to address that one-on-one in the future or in different ways with different people. Okay, how are we? Our poll, the bumblebee is victorious over the cicada and the Madagascar hissing cockroach. So what I'm going to do right now, and can everybody see both my hands and my face? What I'm going to do is I'm going to share some first analog and later digital tools that a scientific illustrator can use. And by the by, whatever I share and talk about, this is just one or a few methods that I have felt comfortable with pursuing or applying. There are 1,001 ways to do any of these things, but let me show you some tools and techniques. So first and almost foremost, a notebook. I love notebooks. I recommend keeping notebooks. So notebooks can be idea books. You can have poetry in there. You can have art ideas. You can collect science data. Notebooks are great. And if you want to organize them carefully, you can have like numbered pages, you can have dated pages, and then everything else is up to you. As far as paper, I like to use the thicker acid-free, and in this case, recycled Bristol board. And so if I flip this over, this is just nice, smooth, rigid Bristol board to draw on. Pencils. There are a lot of different pencils out here. These are H, F, B, all these letters with their respective numbers, 1 through 7, represent different hardness, softness. And so if you've got, I'm just going to use a regular number 2 pencil right now, but if you're really into graphite illustration and you want to play with different degrees of hardness, softness, you can apply pressure that allows you for fine detail or a more sketchy sketch feel to it. So we've got erasers, and I like to use these kind of plastic erasers that leave little erasing nubs on the paper. These are really great. And if you use them and you don't want to erase something big, there are eraser guards, and you can just put it over something and erase that little circle's worth or that line's worth, line segment, right? So eraser guards can be helpful too. Another kind of eraser is a kneaded eraser, and you can form cool sculptures out of it. You can knead it in all kinds of ways, maybe to points. And the idea here is I might dab it on the paper to pick up graphite or rub it just a little bit. These can come in handy as well. We'll be doing pencil, and that's why I asked you to bring a pencil and an eraser. Sometimes it helps to have a brush if you want to brush away the eraser bits. Sometimes if you've drawn, like for example, you've drawn a thick line or shaded a bit. I don't use these, but a lot of people like to use these paper rolled pointy devices that allow shading by rubbing. And then after that, after we do our pencil, what we're going to do together is ink. And you can use all kinds of pens, from a quill pen plucked from your favorite bird, all the way to what I typically use is something like a Zig pen or Micron Pigma pen. And the reason why I use these, because it has a really fine point if it's 005, and they're waterproof and alcohol proof. So we'll talk about that a little bit. I also asked you to bring a ruler. I've got a few here. Ah, I'm back. All right, and my hands are back. Okay. Oh, actually, this is a good time to look at what I have set up here in a big way. So you see what I'm about to do is I'm about to take the winner of our poll, which was the bumblebee, and I'm going to take, say, this bumblebee, and I've got a little helpful thing that I can pin it in and reposition, but you don't need something fancy like this. And I'm going to put it under this dissecting scope or a three-dimensional scope. And the way I'm going to, one guide I'm going to give you to drawing, say, that bumblebee or whatever bilaterally symmetrical object you have in front of you could also be drawn a different way. So this is called a camera lucida. I've got a couple of them here. And sometimes some of you might have a camera lucida as a tube that extends off of a microscope. This is a different kind where, say, I clamp this on my desk like this. Obviously, all I'm going to see is that screen. But say I had a mountain range off in the distance, or I want to draw a forest. Nice thing about a camera lucida is I can stand or sit here, and I've got a half mirror so that I'm seeing not only what's off in the distance, but depending on how much light I have on my hand and on the subject, I see a shadow, a ghostly image projected on my drawing surface. So essentially, I'm tracing what I see in front of me when I look down. Camera lucidas can be really handy. I've used them upon occasion, but actually I prefer to go a different route, and that's the route that we're going to work on today. All right, another tool, and this is a digital tool. You see that I'm staring at this Cintiq. It's a Wacom tablet that's really large, and I can manipulate it by directly pushing things around, right? And drawing, so it's pressure sensitive. There are less expensive ones that you can also draw on, although you look at a different screen, you simultaneously control by drawing on a little tablet. These are becoming more and more common, and I find them useful for everything. So I sometimes jump between using the big Cintiq or something that's more portable like this one. So here's your first challenge, everybody. You have a mirror or you have another human in front of you, okay? I'm going to time you. What I want with your piece of paper and your pencil. Everybody ready? You've got your pencil in hand, paper in front of you. And you either have a mirror and you're looking in that mirror, or you have another human in front of you. I want you to draw that face in front of you in 1 minute. Go.
Apparently some of our attendees are drawing you, Barrett.
They're drawing
And
me.
they're-
Oh my, and I better be looking straight at the screen. That's really important. This could be dangerous, but I have to turn away because I've got to try to turn on back my hands. All right. Look at that face. If it was your fellow human's face, share that human face portrait with that other human. Now, I gave you a really unfair challenge because in a minute or a couple of minutes, to capture the nuances, unless we suffer from prosopagnosia, we can really distinguish faces one from the next very well. We've evolved that way, right? So if I were to look at your drawings, I might see some things like this. So if you can see my hands, I'm just going to scribble here for a moment. So if you drew a face like this, our little head, no, it's just so small, right? The detail will be lost. What you want to do is take advantage of your canvas, right? Is that visible? Oh, good. So you want to take advantage of your canvas. That means like, okay, maybe the face is roughly like this. Every face is different, of course. But if I were to draw a line down the middle, how bilaterally symmetrical is that face, right? Now, it's not going to be perfectly bilaterally symmetrical, but are you way off one way or the other? And where did you put the eyes? So if you go roughly halfway down, strangely, of course, it varies face to face. That's where the eyes are. Imagine putting in the hair there, right? And then halfway down, maybe that's where the nose goes. So the idea here, and halfway down, maybe that's where the mouth goes. When I'm drawing, when I'm sketching something, I might be losing the proportionality. But if I were to carefully measure, and this is why you see artists holding up their thumb or maybe a pencil or a paintbrush or something and going up and down like this, they're measuring one thing relative to another, and they're trying to match that on their canvas. That's our goal with our technical or scientific illustration. Okay, so put your face away. And now what I want you to do is grab, and I hope you got this message early on when you registered. I want you to grab something that you'd like to draw. So something that excites you. It can be small, it can be huge, and preferably something that has a mirror image of itself, something that's bilaterally symmetrical. Doesn't matter if it isn't, but some of the steps I'll be taking take advantage of the idea of this bilateral symmetry. At least to start the drawing, this is what I'm going to do. Actually, you know what? I'm going to ditch that for the moment, and I'm going to show you with my hands what I'm doing. So here's my bumblebee. Here's my drawing board. And what I want to do is not draw this actual size because I'll have a tiny bumblebee. And if we want to publish this image later, oh, hey, look at that, a face. That's great. So someone's able to post. Awesome. So what you want to do is make something large, right? So that if you wanted to publish it, you could shrink it down. And shrinking it down tightens an image. So even if you think it looks a little sloppy at actual size, it'll end up looking a lot better when it's significantly smaller. That said, if we make it really detailed, you'll lose the detail if you go from huge to tiny. So we want to find that balance. And here's how I recommend approaching it. Here's one way of approaching it. Let's say this is your canvas. I'm going to take a ruler, and I'm just going to draw a really light line down the center. Great. I drew a line that you probably can't see on the screen, but it's there. There it is. You know what? I'll just artificially draw it really dark. There's a line down the center, okay? And what I want to do is measure, and I'll use calipers, but again, you can use a ruler. And with these calipers, I'm going to say, all right, how long is my total organism? Now, this would be a lot easier to do through the microscope. But I'm going to say, all right, from this angle, my bumblebee is 1.9, let's say it's 2 centimeters long. Now, I could draw it 2 centimeters long. That's really tiny. But let's say I want to make it 5 times bigger or 10 times bigger. If I make it, I'm going to make it 5 times bigger. Then it's 10 centimeters long. Ding. And you see how I'm making a little, very light, in your case, very little light marks there. Sometimes I'll make it much larger than this, but for now, I'll just make it 5 times bigger. Okay, that's only one measurement. Now, let's say I want to take the measurement of that big midsection, the mesothorax. Now that is 1 centimeter. It's actually half of the length. So the top part, the head is kind of hidden. It's only under here at the angle I'm looking at. So I'm going to go half the length, which is 5 centimeters down. That means this whole thing is going to be that midsection. You probably wouldn't guess it if you were just sketching it. By taking measurements, I'm really getting that down. So that means I've got close to 1 centimeter for the abdomen or the metazoma here. Okay, these are only a couple of measurements. Let's do more. Let's do the width of various parts, like the widest part of that metazoma or the third segment of the body. And that's 1.1. Now, what am I doing here? I'm taking every single measurement from the real organism and I'm scaling it up, in my case, by 5. Maybe you're doing it by 10. Maybe you chose a giant subject. If you did, then you're scaling it down. So if it's, for example, 1 meter long and your paper's only so big, maybe you're going to go 1/10 the size or 1/20 the size. But whatever measurement scaling up or down that you choose, you have to do all measurements up or down. So say, for example, my width of the mesosoma, the midsection of the body, ends up being this wide. I measure half the distance here, and then the other half there. So it's bilaterally symmetrical. And let's say the width of the wings are at this point. And let's say the widest measurement of the mesosoma or the metazoma, the final segment of the body, is here. And let's say the head, which we barely see, is here. And let's say, and you keep doing it so that, say, for example, you did 1 million dots or 1 million measurements, you'd be like Seurat, a pointillist, and you'd have your illustration. Now, we're not going to do 1 million dots. We're not going to do 1 million measurements. But if you do a few key ones and you keep adding, then you can feel more comfortable about approaching or approximating your subject with correct, anatomically correct, proportionally accurate measurements. For participants, say in my scientific visualization class, they did this beautifully yesterday. One wasn't so patient and said, you know, I really hate doing these dots and these measurements, even though it only takes a few minutes. If you jump past that and you just start drawing as you think it should look, you might have some shocking surprises by the end where your insect, in this case, is way off the mark. So again, I'm taking measurement, 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 length and width, both to the left and to the right, or up and down, depending on what measurements I'm taking. And then when I've got that down, when I've got those different dots down, I can start to connect those dots really lightly with pencil. And that's what I want you to do, really lightly. So what I've done here is I made that line because I'm looking at a bilaterally symmetrical organism. But even if you're not, if you're taking careful measurements, then one thing relative to the next should play out. Now, I'm going to back up and I'm going to remind you that there are different ways of doing this bilaterally symmetrical organism drawing. One is with the camera lucida, where you're essentially tracing. Another is, if you wanted to, you could create a grid. You measure to the left, to the right, to the top, the bottom, and you create this careful grid. And let's say you have a grid in one of your oculars in your microscope, or you even put a grid in front of you. So the landscape you see or whatever you see has that grid. Then you go square by square. That's another approach. So there are a lot of different ways of doing this. But let's say you're continuing with this and you're making your measurements left and the right, and you start doing this rough outline and you see that insect being formed. Let me pull it. And I make the measurement here and I see that wing comes out like this and hind wing is like this. And I see the other wing comes out like this, the other hind wing. And obviously I'm not doing this carefully and through the scope, I'm kind of rushing this to give you an idea. Here are the elbowed antennae, and I make measurements to make sure how long they are. Here are the compound eyes, right? And gradually I'll get the legs. I see bits of the legs apparent, you know? And then I'll start filling in. I see a segments here and other segments there. I see this area is going to be dark. And I see this area is going to be really fuzzy with this dark area here, right? What I'd like for you to do is take a little bit of time, go through and make the measurements, essentially round out roughly connecting those dots so that you have a clean, clear illustration that proportionally is accurate, that matches your subject well. If you're starting to feel really comfortable with it, if it's looking like, yeah, that's matching my bumblebee, right? Then what I want you to do is start making harder lines. Like, yes, I'm pretty positive about this part. And when you've done that with your pencil, with the harder lines all around, right? I'm feeling pretty comfortable here. We're getting toward the point where now we could either ink it or, and I'd want to add a lot of venation here and the wings, right? I'm either going to ink it or we're going to do something different. If you chose a bilaterally symmetrical organism, I want you to choose one half, the left half or the right half. And I want you to do a line drawing, which means basically just the hard lines, the outlines of the different features without much detail. A simple line drawing. The other half will do something different. And that different is to make it three-dimensional. And I'll show you some tricks in pencil, in ink, to make this a more realistic, the right half, a highly realistic three-dimensional rendering. So how about you take, let's see where we are in 10 minutes. At the end of 10 minutes, I'll give some techniques for three-dimensionality, and then you can have your hand at that, and then we'll do something very different, okay? Again, what did I do? I drew a line down the center. If I'm thinking bilateral symmetry, I'll make it dark again so you can see it better. I make careful measurements, and this will take much time. If I, especially through the microscope, where it could be even more helpful, I'll find that linear measurements along the body axis, as well as away from the central axis, I can get left and right, up and down measurements. And as I make those measurements, I find that it helps to do the whole measurement of the insect. Let's say I do the bumblebee this long. Now I have done what? Taken 2 centimeters and made it 20 centimeters times 10. Hey, there's somebody's great. Thanks for posting. So if I've magnified by 10, then again, I'm going to magnify everything by 10, okay? So I get that shape as I do more and more dots, right? More and more measurements. Now, what I want you to do, if you feel like you're right there where you want to be with your line drawing of your subject, there are a few things we're going to add, and we're going to do this mostly digitally, but I want you to get a feel for what you're going to add right now. A figure legend. Figure 1. What's important to put down here? I'm going to put down that I've got a dorsal view, that means the view of the back, dorsal view of, and let's just say female bumblebee. And in parentheses, I'll put Bombus species. and illustrated by or illus, and you can put your name. I'm putting B. Klein, 2024. Give yourself credit. Okay, so you've got a figure legend. We'll redo this so it's typed and beautiful. If you want, you can point out anatomical features. That's a compound eye, that's an antenna, that's a forewing, that's a, and this is something we can type out too. So you'll have little labels typed out here, 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 and here. The last thing that I'd like for you to include in your figure, which is really, really important, is a scale bar. How big is this bumblebee? Is it the size of your thumbnail or a billboard? We don't know unless we have some kind of scale, some kind of relative scale. Now, a scale bar, and I can put it horizontally here, or I can put it vertically there, doesn't matter. I'm going to put some measurement so that I can say, all right, this little bit, I see now how long this wing is, or the abdomen, or so a scale bar can be really helpful. How do you do the scale bar? Well, let's say, for example, remember my real butterfly was 2 centimeters long, but I made the drawing 10 centimeters long. So I multiplied every measurement by 10 or by 5. I want to do the same thing with a scale bar. So if you look at this bumblebee, I want to make the scale bar maybe half the length or a third the length or a fifth the length, something smaller than the bumblebee and a nice round number. If she's 2 centimeters long, and I'll draw her up here just for yucks. So here's our bumblebee, right? And she's 2 centimeters long. Now I want the scale bar to be that long, half the length of her body. I want it to be 1 centimeter. Since I multiplied this bumblebee times 5, I'm going to multiply the length, that 1 centimeter that I want the scale bar to represent, times 5. So this will now be 5 centimeters long. What number do I put down here? If I put 5 centimeters, because that's the length of what I made this, think of how big the bee would be. That'd be a huge bee.
There
So
we're getting answers in the chat of 1 centimeters and 2.5 centimeters.
Rock on. You want to write 1 centimeter because everything is being scaled up visibly here, including what you want to make your scale bar. But what does that scale bar represent? It represents 1 centimeter. So you'll put 1 centimeter down there if it is 1 centimeter. Okay. If you want to think about this in terms of mathematics or an equation, then you just say, all right, what is the length of the bumblebee? The length of your subject, or any part of your subject, length of your subject divided by the length of the drawing of your subject. And in this case, it's 2.5 over 10, or 1 to 5, 1/5. That better equal 1/5. What's on the other side of this equation? What you want your scale bar to be divided by what the actual length of the scale bar is. And we know that the actual length is 5 centimeters. So the x in this equation, 1 over 5 equals x over 5. That x equals 1 centimeter. So if it helps for you to think about it mathematically or in terms of an equation, you can do it that way, because sometimes it gets more complicated numerically than what I've presented here. But you can also think of it intuitively, like I scaled up this scale bar, but I don't want to change the number because this represents 1 centimeter. Because, well, let's test it out. All right, I'm going to take my ruler. If this represents 1 centimeter, it's actually 5, right? Then if that 1 centimeter is put over here, then I see, great, that means my bumblebee is 2 centimeters long. Bink, bink, right? So it works. So when your drawing is ready with your sketch, your line drawing, with your figure legend, your scale bar, and maybe pointed to some features, now we can go to the next step, unless I have any key questions right now.
When you're drawing through a microscope, how do you take measurements? Do you use one of those special oculars with a ruler on
There
them?
are a lot of different ways to do it. You can do it that way. If you've got an ocular insert there, that's either a grid or actual measurements, or you can use special, depends on the scale of your subject. Like I've used some where I drew something so tiny that I actually had, I forgot what they're called, but they're inscribed in metal, the tiniest of tiny measurements that you put under the scope beside your subject. That's one way to do it. Or as I was doing with my bumblebee, I just take these calipers and do the measurements under the scope. And that's so much easier because it's magnified and you can see very clearly each and every measurement.
Few people are asking if you could do the scale bar measurements one more time.
Oh, certainly. And by the by, in my in that link within the SciViz links sheet that I'm making available, there's a link to a newsletter, step-by-step guidelines that goes through this very clearly as well. But here, I'll do it. I just did a bumblebee or whatever this is, right? I did this thing. And let's say the original looked like that. And the original is 1 centimeter long. But I want to make it big. I want to make it 20 times bigger. So I measure 20 centimeters. And I find that this part is 2 centimeters, and this part is 8 more centimeters, and this part is 10 centimeters, right? So I'm doing these measurements, I'm doing all these other measurements. Now, scale bar. If this whole thing is 1 centimeter long, I'm going to make my scale bar, let's say half that, 0.5 centimeters, or I'll just say 5 millimeters. That's how long I want my scale bar to be. Something smaller than the original. And I'll say 5 millimeters to 10 millimeters for the whole thing. Okay. Half the length of the whole thing in this case. Now, remember, I scaled everything in my drawing up by 20 times. So I'm going to take that 5 millimeters, or maybe it's easier to look at it in centimeters here because it's half a centimeter, 0.5 centimeters. I'm going to multiply that by 20. Well, times 10 is 5 centimeters, times 20, right, is double that. So 5, so it would be 10 centimeters. So if I put my, let's say I put my scale bar like that, and I say 10 centimeters, something's wrong here. Now, this is right. I made this 10 centimeters, and this is right because it's 20 times the length of the scale bar I'm aiming for. But if I say just 10 centimeters because everything else was scaled up 20 times, look how big my drawing of my bee is. Look how big my bee is. It's 20 centimeters long. That's a huge bee. So while I scaled up 20 times, my scale bar represents that number, 0.5 centimeters. And again, if you want to see that as an equation, I'm going to say my bee is 1 centimeter. My drawing of my bee is 20 centimeters. 1 to 20 should equal what I want my scale bar to be over the actual length that I draw it. Well, what I want it to be is 0.5 centimeters. And I'm putting the X in the denominator because I'm doing it the opposite way now. But how long do I draw my scale bar? Well, let's see. 20 times 0.5. So 1x equals 10 centimeters. 20 times a half is 10. So that means x equals 10 centimeters. And sure enough, that's how long I would draw this. But the scale bar, remember that I'm desiring is 0.5 centimeter, because that's what I wanted next to the original. I hope that's clear. Let me know if anybody has questions. I'm going to jump in to part 2, and this will be pretty fast, but I want to share a couple of techniques. And by the way, if you are interested, there are lots of resources out there. I've got a whole pile of books behind me, but this is the go-to, the Guild Handbook of Scientific Illustration. Get it out of your library or interlibrary loan. It's a wonderful book by the Guild for Scientific, Natural and Scientific Illustrators. That's the best resource. And that's, again, in my links, where you can go online and learn so much. But in this book, they also have really beautiful examples of how to approach scientific illustration. I want to make something 3-dimensional. Now, I could pull out my colored pencils, and we could do it in color, or I could do it in paints. There are endless ways we could do it, but what I'm going to pretend is that we're going to work in ink. So you want to publish something that's black and white. Well, how do you make something 3-dimensional if it's two-tone or black and white? Well, it's fun and easy. So for example, if I've got a ball, a sphere, right? Is this a golf ball with dimples, or is it a ball with little bumps? Who knows? The best way to tell is with lighting. So if light is coming from this angle, like I'll pretend this other circle's the sun. So say that's your light source. The sun is striking your ball. You would expect a shadow to be here, right? But you'd also expect shadows in here. So if it's dark within these little circles, then they're dimples because the light isn't able to get in there. But if they're bumps, then you'd expect the outer part of the bump to be darker and to cast a little shadow. We can use that principle plus, like, say I've got a pipe, a metal pipe, and light is coming from here. Well, if it's glabrous, if it's smooth, if it's metallic, the light is going to be bright. And maybe it hits this part right here, which means you're going to have this dark area here. And maybe a slightly dark area up here to give that rounding effect. If I make it dark right to the edge, then it begins to look kind of flat. If I didn't have the darkness up here and a little bit of light to show reflection of light back, then it ends up looking flatter. Similarly, if I've got my sphere here, oh, let me do another one. If I've got a pseudosphere here, then what can I do? I can take my pencil and I can kind of shade in, right? If I'm doing a pencil. But try doing that in ink. That doesn't work very well. So instead, I have to think two-tone, or I actually do it with ink right away. Let's pretend this is my pen. And what I'm going to do, actually, maybe I'll get a thicker pen to give an idea. Have it right here. So we'll just start with a soft pencil. And one way that you can get this 3-dimensional effect, let's say the light source is over here, is to think in terms of this being bright and light. If this is a smooth ball, this area is going to be bright and you won't see any darkness at all. As you get away from the light, it's dark, like really dark, potentially, depending on how striking the light is, right? Now, again, just like that metal tube, notice that I'm not going right to the edge, right? Maybe I'm giving a hint that it's not pure black on the edge. And the reason for that, again, is that you can have some back reflectance, and it also helps round out the ball. Over here, I'll have a little bit of darkness, and I'll continue on. Now, again, how do I do this in ink? Well, if I do dots, what's called stippling, and I love stippling so much that at a layover in Nevada, Las Vegas, I stippled right through my flight, so I missed it. I slept in a doorway in the airport until a security guard kicked me. So stippling can be a great thing or a wicked thing. I love it. So as you're stippling, like from a dark area to a light area, you see a gradation. Everything's in dots. If you squint, it becomes smooth. The more dots, the smoother the gradation of dots, right? Until maybe you have some dots here to round it out, right? So the more dots, and let's say I've done this for an hour and I've got lots and lots and lots and lots of dots. And yes, you can do this digitally. It's not as fun. You end up getting a really stippled area. And obviously I'm rushing it, but the idea is you're getting these separated dots representing lighter areas. Let's say it's not a super smooth ball, so you don't have this bright area here that's untouched by any dots. Well, if it's something that's like, if it's texture, if it's fabric or something like that, that's when you can start putting in a little texture. If, for example, it's dimpled, well, there you go. In those bright areas, you can have those dark dots. While in the dark area, you can have light dots. That shows where the light is hitting, say, bumps or dimples. And by playing with that effect, you can get great three-dimensionality, okay? So stippling is one way, all the series of dots, right? The thicker or more dense the dots, the darker, the more sparsely placed, the lighter. Another way is to do hash marks. So let's say I'm going like this. What if I cross over like that? All of a sudden, this becomes a darker area. What if I keep crossing over at a different angle? Again, this becomes darker and darker and darker relative to the other lighter areas. You can do the same thing for your sphere with hash marks, with stipples. Another way to do it is, let's see if I see how I'm doing like an old style etching. You can have thicker lines to lighter lines, and you can do this digitally as well. So there are lots of different ways of getting that three-dimensional effect. If we go back to our bumblebee, this is really hairy. There isn't going to be too much smooth. So the light effects that I can show will be a little bit different in something that's matte. Now, I can show that in the compound eye, where it gets dark on this side of the eye, but lighter on the other side. So this is away from the light if the light is coming from here. Again, I don't go right up to the edge, but this is the smoothest part of this bumblebee. So this can be bright, those compound eyes, for example. Again, it'll be darker away from the light. This will be darker too. So maybe when I show the hairy parts here, I can make it a little bit darker as well. Just a little bit, hints of that. Now in my bumblebee, this is a dark region. And if I look at it under the microscope, yeah, it's still pretty hairy, but I see that there's some shininess in here. So I'll make it really dark over here in that dark patch, but I'll leave open little white dots that lead to the bright area where I have a few dark dots. And that tells me that there are dimples in there. Maybe it'll help when I show final illustrations and when we play with this digitally. Daria, could you take an Insti poll?
Yes.
Find out if people would like to see stepwise progression of examples of digital illustrations I've done, or if they want to jump right into me working on their illustrations digitally. All right, I'm going to end this poll now. It's pretty obvious that I'm going to talk about digital media because we got 379, 386 to 40 at this point. Okay, I'm ending the poll. And what we're going to do is talk about two different formats or approaches to creating digital illustrations. Notice the top one is rasterized images, and the lower one, vector-based images. And you see the little Smithsonian Institute's logo of that weekly ray of sun. The top one, if I blow up a rasterized image, it'll be blurry. Why? Because if we're thinking digitally, it's made of pixels and made of only so many pixels. So say it's made of a bunch of pixels and you try to put something that's on that person's shirt on a billboard, it'll degrade. There are too few pixels to have a high enough resolution there. Rasterized images are images that you can't make infinitely large. Programs that are raster based include Adobe Photoshop, but if you're not at an institution that pays for that, or it's too expensive for you, then I recommend using GIMP or some other freely available program, which is also really powerful. So go GIMP unless you have Adobe Photoshop available. Those work with, say, for example, a photograph or the drawing you just produced. Versus, see that little Smithsonian symbol on the bottom? Now you blow it up huge, infinitely large, it'll retain the same resolution. Why? It's based on vectors. So it basically recalculates all the vectors, no matter how big you make it, and recreates it. That'll be Adobe Illustrator or the freely available Inkscape. I'll be using both Photoshop and Illustrator, but I could use GIMP and Inkscape equally smoothly to alter the visuals we produce. So this is an illustration I did back in 2010 for to show that Insominator, all those magnets that I'd roll across. Here are 2 frames of honeycomb, 1, 2. And I'd roll this bank of magnets across to wiggle those bees with Ferris tags on their backs. This became really helpful because a photograph is really nice, and I included a photograph in different ways, but this lays out, okay, what materials were used and where do they go? And looking at it proportionally as a blueprint really helps, increases the replicability. Now, this is produced almost totally using a vector-based program. So you can scale this skeleted bark beetle up and down. This is one I did with my mom that looks at a morphing from a bat wing to a human hand. To think about homology, our finger bones, shared common ancestry. This is the cover for a field guide I illustrated, Damselflies of Texas, with John Abbott. And this is a very different approach, but we took dead damselflies and I disassembled them, scanned the individual parts, and then digitally, everything else, reassembled them, cleaned them up, painted them essentially, sometimes with many layers, 50 layers for a single damselfly. So the whole book, species descriptions, distinguishing them in, I think it was something like 90 species or so. So it was a lot. And the value of working digitally in this way for me was that John Abbott could say, that's okay, but on average, that species has more pruinosity or is waxier or is a little bit more yellow. And I could go to a layer in Photoshop and immediately change that. That's powerful. So they're versatile programs. So for example, here would be a screen I'm working with, and these are species I'm working with. And I'll pull colors from photographs taken in the field of the damselflies. And I'll lay those colors down. And I can, if we have time, I can show you exactly how I do that. Here are the pieces that I'd scan, cleaning up those pieces digitally. And notice I had to scan each part at the right angle. And notice that that abdomen lateral view right here is all curved. Notice that the head is brown and sometimes the eyes are shriveled. They look dead. But to create them in this fashion on the right, what did I do? I pulled colors from photographs, these little things here, 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 so that I could show examples of males and females. So these ended up being the final for this particular male of this particular species of damselfly. I defy you to try to take a clearer photo in the field of a darning needle. They're really hard to photograph, but having them on a white background where you can really play with the detail of the venation and everything helps. Remember I mentioned that ant that my friend Christian Robeling discovered, Martialis heureka. This is the only specimen in any collection in the world, and it helped people understand how ants evolved. There it is, only 3 legs, because the other 3 legs were ground up for a genetic assay. And this marvelous little ant, Christian said, hey, can you draw it? And we overlapped for 24 hours, and within that 24 hours, what I did was I took notes and I sketched out what he was looking for, right? And what I saw through the microscope in this single valuable specimen. I took the sketch and I drew it a little bit more like we're drawing it, taking all the measurements and everything. And I decided to scan that in. And then I added layers from textures and photographs that I manipulated. So it looks kind of like, there it is, burrowing. He didn't find it underground. This 3 millimeter long ant bumped into his foot, right? In the leaf litter of Manaus, Brazil. But he said everything about it, pale in color, eyeless, suggests that it's fossorial, that it's burrowing. Can you show that? Note the scale bar. You might think this ant is big until you put that scale bar against the ant to see, oh, it really is only 3 millimeters long. Fred Dyer took this photo of a honeybee gobbiting, and that's basically regurgitating and holding this water droplet that a honeybee can use to cool down the hive or the nest, basically by fanning wings and evaporative cooling. Well, Tom Seeley wanted me to display this visually in his book. I looked at a bunch of dead bees that I had in the lab, and I kind of dissected them and I pulled the mouth parts out because I pulled them from the freezer. So I could really look at their mouth parts carefully. And I looked at drawings in the literature and tried to figure out the anatomy and tried to extrapolate to better understand how they'd hold this droplet with their mouth parts. And with all those images, and you note in the middle here, I actually built from a photograph. I said, okay, I'm going to sketch this at first. So I've got some of the proportions down. So here's my drawing, right? Here's my rough drawing of the gobbiting bee. And here's the kind of finer drawing. So I always start with a drawing. And then I scanned it in and I digitally superimposed some color and texture, and things like that. And Tom Seeley and I looked at that and said, well, that looks like an alien. It definitely needs hair. So at first, we wanted to put it in the hive, in the dark hive, to show how it's used in the nest. So I put a photo that I blurred out of honeycomb in the background, started shading with digital layers over the bee to get some 3-dimensionality. And then I started, see now how blurry the comb is in the background? So your focus is on the bee in the foreground. And then I drew with pressure-sensitive tablet the different thicknesses of the different types of hair-like seedy all over, even the compound eyes, all over the body. And you note down here, whoa, there's a shiny part on this droplet of water. There's a shiny part along the leg. All I did there was I created a new layer in Photoshop. I drew a little white, like painted a little white, and then I blurred it out, just shaking over it in a larger smudge tool, blurred it out so that it could get that effect. And I think lastly, I'll share how I created an illustration that shows the twitching, curled legs of a sleeping, potentially REM sleeping jumping spider. So here's a video that Daniela Rüssler took of these jumping spiders. See these twitches? So weird. Well, you can even see their retinas just darting about within their prosoma, the front part of the body. Well, I wanted to capitalize on the idea that nobody's ever looked at spiders with respect to REM sleep. Nobody's looked at arthropods. Nobody's looked at invertebrates save for an octopus, and only behaviorally. Now, if you look at a mammal, if you look at reptiles like this bird, this lizard, people have measured brain waves to look at rapid eye movement sleep. But octopuses, not quite. And then if this is a phylogenetic tree with the most recent common ancestor here over 500 million years ago, invertebrates and vertebrates, or vertebrates popped out of the invertebrates, you've got this octopus and then this spider. So I did this drawing, cleaned it, scanned it, cleaned it up digitally a little bit. And then I started thinking, oh, wouldn't it be fun to have a moon as a backdrop? So I took a photo of a moon and I kind of changed it around a little bit. And then I selected areas and I filled in those areas. So I wanted a deep blue. And then I wanted a black from above and dots that represented stars. And then I took the real electroencephalogram readings from a sleeping mammal, this bird, that lizard, those species that were published. And then I made them, scaled them appropriately for amplitude and the rest, and then superimposed them with color where they were appropriate. And then you've got this phylogenetic tree that gives you a picture of how different lineages sleep with the potential that spiders engage in rapid eye movement sleep. In the guidelines I give in that tutorial, I took a damaged yellow paper wasp, Polistes flavus, from Arizona. And I took those caliper measurements, and there are the lengths and the widths. A lot of them in this case. A rough drawing to see how it would fit and how it feels. And then the finer illustration. I think about the scale bar, and there's that equation I talked about, measurement of or on the organism over the measurement on the drawing should equal the scale bar label over the actual scale bar length on the drawing. And there's my URL for that tutorial. And so my drawing then, I can point to anatomical features with a scale bar. And there is two-tone, that's black and white line drawing with certain level of detail. Now I could make that in color too, right? And give some three-dimensionality in a very different way. Hey, 95 of you were brave enough to share your bilaterally symmetrical drawing. I'm impressed and grateful. So my goal here right now is to take several of your drawings, and I'm going to show you what you can do in Photoshop, a raster-based program, and then in Illustrator, a vector-based program. Okay? And what I'd like for you guys to do is work along with me. But wait, it's time for a tea break. And during this 5 to maybe 7-minute tea break, certainly ask questions if you like. If you happen to have Photoshop or GIMP, Illustrator or Inkscape, pull those up and take your drawing and follow along with me. If you don't happen to have access to those programs right now, just follow along and ask questions as you have them. Hey, everybody. So I am loading up the images, and while they're loading, if you have any questions, I'd be happy to address them.
There was a question about the use of biorender in scientific papers now. What do you think of that?
One philosophy is that whatever works, go for it. As long as you're being responsible, respectful in terms of acquisition of source material, and you are giving proper uses and citing sources, and you get proper permissions, biorender is an exceptional thing where I think it's all freely available. I've seen examples where they've worked quite well. I've seen examples where liberties are taken. For example, that's a frog, right? But it's the wrong genus of frog. Inaccuracies can be problematic, and biorender is kind of like clip art in a way that some things can be quite simplified. So you just want to think responsibly and carefully. But again, if it works for you, if it conveys ideas effectively, visually, in a responsible way, great. This is a good opportunity for me to say, if you're joining the second workshop, please bring examples where you've used anything, including biorender, and we can talk about whether or not that image use and that biorender use is appropriate, because sometimes it's a case-by-case situation.
There was also a question about resources for converting raster file to vector file.
You can't do it. Well, I should take that back. You can't just change raster-based to vector-based, but you can trace your raster-based image, so then it becomes a vector-based image. So for example, you've got a photograph of a sperm whale, and you want to make a beautiful logo or a poster of that sperm whale. Well, then you can use, and we could, if we have time, we could show how to do that. You basically just take that raster-based image, that photograph, as one layer, as your background layer, and then work with another layer over that, where you then start using a pen tool to start tracing curved lines to make that sperm whale perfect. And you can use colors, you can add detail in layers, and actually that de novo ground up vector-based art proficiency, I highly recommend joining the workshop with reaching a broader audience with Damon Kylo, because that's his bread and butter. He's so proficient with vector-based imaging that he'll be your hero there. Any other questions?
There was one other one, I think maybe lots of people would be interested to know the answer. What are your general best practices when working with many layers?
Oh, yeah. So for those who don't know what we're talking about here, layers, I'll show you what I mean when I pull a couple of your drawings into Photoshop. The idea here is, if you have just one layer, your background layer, that's your image of a bumblebee, and you start drawing on that, and then you add something into it, right? And you always maintain one layer. What you've drawn on top, you can't change without changing everything underneath around it, because now it's permanently fixed to that single layer. But the power and versatility of having multiple layers is that you can just take one layer and say, I'm going to change the hue of that, or the contrast, the opacity, or I'm going to eliminate that layer altogether, or edit it with an erase tool. Wow, that's so much easier than trying to work from scratch. And that's why I did the Damselfly field guide as a Photoshop kind of factory lineup series of digital illustrations in that way, because I had that versatility. Now, best practices means while Photoshop allows you to right click and select a layer just by tapping on it, for example, sometimes it can get a little tricky. So labeling your layers, having the list of layers out there in your tools and labeling them appropriately can be really helpful. I hope that helps. If there's another deeper question about that, let me know.
Here's
Any
another
other?
interesting one. If a researcher wanted to engage artists for the work, where would they find resources about appropriate compensation, how to include scientific illustration in the grant application, etc.?
I do that all the time. So for example, for the book I just finished and the book I'm working on now, I hunt down the source. You can't cut corners there. You have to find the original source. If it's not obvious, like you haven't found it published somewhere, or if someone hasn't responsibly labeled it online, like in Pinterest or something like that, you have to hunt it down yourself. One way to do that is to drag it into Google Images to see what images match. Then you select those images or the URL associated with those images and see if they were responsible enough in giving citations or attributions. Oftentimes I'll do extensive hunts for photographs or illustrations. And I can give you case-by-case scenarios here, but for example, one, I hunted for a beautiful photo that conveyed an idea really exquisitely for the sleep book. And I found that the attributions varied. And one I found out was absolutely incorrect. And there was a bit of a scandal there, but I hunted down and say through Flickr, and then I found a way to contact the original photographer and wrote to that photographer. That's really the way. Many things are freely available as Creative Commons, Wikimedia, for example, and you can hunt for images that way. But even then, I'll cite the source, and sometimes even then, if they're living, I'll go to the source and communicate with the source.
There are other questions. Do you want me to keep going, or do you want to move on and then we'll get the questions to you later and get the answers to people later?
How about pick one zinger?
Oh, zinger.
Yeah, real zinger.
There's so many, like,
Your personal favorite, Joanna.
well, the one that's on my screen right now is, are there principles for what goes in a single layer? When drawing digitally, how many elements go in a single layer? Is there some sort of rule of thumb?
Good question. If you don't want to be inundated with hundreds or thousands of layers, yes, you'll want to start combining things. But the more you can separate, the more powerful or versatile your stepwise progression in your image is. Oftentimes that's a case-by-case scenario. Like for example, I want to work with all black or everything involving the wings in one layer. But if I did that for white, when I like to smudge out to get that blurry effect that you've got a reflection off of a surface, if I did that all in one layer, I'd be in trouble. Because, for example, in a wing, I might want to do white there, 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 there. When I smudge one, it'll smudge the others. So now all of a sudden, I've got layer after layer after layer. So it really depends on what works for you under specific circumstances. And we can talk about case studies to consolidate or to split up. But splitting up, as long as it doesn't become overbearing and overwhelming, is usually the way to go, just not to ridiculous degrees. What I'll do is I'll just choose these few for now. And if we have time, I'd love to go through more of these. This is great. How about, I know, let's take a poll. What do you want to see me fiddle with first? Rosalind, human skull, other skull, kettle, rose, or one of these two bumblebees? Well, it looks like a solid third is going for other skull. So let's do that. So we've got this skull, we've got Rosalind Franklin, we've got the tea kettle, which is really beautifully done. I want to know who this person is, reach out to me. And oh, that's so cool. Then there's this, and there's that. So we're looking at this one. This got the vote. Okay, so what do I want to do first? What I recommend doing if you're new to a raster-based or vector-based program is play with one tool at a time. And I am not really proficient at keystroke efficient methods. There are, again, many different ways to accomplish the same thing. And many of you out there will do this far more efficiently and in many different ways than I, but I'll stumble through it the way I'm used to, knowing and trying to convey that I constantly use Google searches and YouTube video tutorials whenever I want to use a new tool or a tool in a different way, okay? Other skull still wins. What I want to do is I just want to grab some of this, and maybe I'll grab with this. Don't be overwhelmed if you don't know these tools, but I'm going to grab this tool, which is basically, it's called a polygonal lasso tool. And if I want to grab boink to boink to boink to boink, right? Now I'll cut that X and I'll paste it. So there it is. Maybe I'll hide. See how I hit this hide in the layers here? I'm going to start building up layers, which is cool. So I'm going to hide that background layer. Now it's on white. Let's say I'll take, it's under Edit, Transform, or Free Transform. The other way you can do that is Command T. And I'm going to click this rotate, like on one side, and rotate it, right? I can do it this way, but I might want first to change my canvas size. So under Image, I've got canvas size, and I'm going to make sure it's in the middle here, not off to the side somewhere, but in the middle. And I'll make this taller. And the background is white. You see here there's a brownish, here there's a white. That tells me the background will be white. I hit OK. Now it's bigger. I'll zoom out a little bit. And sure enough, it fits better. I'm going to hit that Command T again. I'm going to rotate. Now, what if I can't get it quite at a 90-degree angle? I can't turn it quite. Well, if I hit Shift, then it goes in increments. Great. Now, what if it's not exactly where I want? Well, then tweak it a little bit. There. This is on a separate layer. It's the top layer. So I can move it around. Now, ultimately, I can hit this and crop the whole thing so it doesn't go crazy big. And, you know, maybe I'll want it something like that. Okay, so how do I want to change this? Now, this is a hard image to work with because it's really grayscale. I had you photograph because that's really easy. The easier approach to work with is if you scanned it. If you have it in a scanner and it's, say, black on white, then it's way easier to work with than, say, this dark gray versus this light gray. But we'll see what we can do. Okay, first, I want to create a figure legend and a scale bar anew. Right, that's what we'll do. But before we do that, how about I just at least try to make this more two-tone or white? How do I do that? Well, if I go up to Edit, Image, and I go, well, Mode, it's in RGB color. Do I want it grayscale? Will that make a difference? Maybe not. Maybe I want to add color. But see, if I do grayscale, it'll ask me to flatten the image. I could do that, and then it'd be purely grayscale. But let's forget that for now. Image adjustments. I'm going to look at levels. Now, you could do brightness contrast. If I do brightness contrast, I can make it bright. Oops. I can make it. Let's go. Right. Or higher contrast, right? That's one way to go. Maybe you're getting it closer to what you want. Or image adjustments, levels, I really like. There are different ways, exposure curves. Levels is kind of nice because if you look at these levels, there's no use here. There's nothing under the curve past this point. So I may as well bring it in there. This, I don't think it makes much of a difference either way, but the middle part I can start working with. So it's at least a little bit easier to work with than the contrast brightness alone. Okay. Now, again, there are lots of ways to do this. What if I made the front white and I use the bucket tool and dumped white in there? Does that help? Probably not too much, but maybe it does, right? You know, there are lots of different approaches. No, but then you get this hairiness. So yes, a bunch of you out there know how to do this far better than I do with masking and the rest. So let's get back to where we were and pretend that it's okay. What I want to do right now is just play with the image a little bit, okay? And if I decide that I want to have at least the outside whiter and make it look like a cool gray skull inside, but that's not enough, maybe I need to hit the erase tool, and I can make the erase tool big or small. I can make the erase tool hard-edged or soft-edged. And I can go in there, I'll make it a little bit bigger, and erase this other stuff here. Now, that's one way to do it. You can just erase around it, or you could choose magnetic lasso and travel around something and select an area and fill that in or erase that altogether. There are lots of different approaches you can take. But let's say, hey, this crazy skull, whatever I did to it, all I want to do now is add some paint. And I'm going to add some hot pink, right? And maybe I even want to add the hot pink in the eyes. Or, oops, or I want to mark a spot. Like this is especially important to me. And there are a lot of things, well, just don't have the time. But let's say, right, this feature is key. And I could make a dot right there, right? So I can paint on it, I can erase it, I can make blocks of what? I can do all kinds of things, right? Limitless for a raster-based image. Note though, failure, everything's on one layer. Now I can't fiddle with this red dot. I can't move that red dot. Let me undo that. I'm going to recreate the red dot, but now I'm going to create a new layer. So layer, new layer. And I'll name it red dot. So see, now it's at the top. That's great. I'll put my red dot right here. Oops, I'm on it. And I've created my red dot. Okay, great. I can hide that red dot. I can move that red dot. Power of layers. And we could play with filters. We could keep going. What I'm going to do instead right now is say, this is exactly how this person wanted their skull to look. And now we'll say, I've done enough with raster-based stuff. I want to pull this in Illustrator, and I want to have a really clean typed figure legend and a really clean scale bar. File, save as. Really key message for everybody. Please save whatever project you're working on as a raw file. You just opened it up. You haven't done any changes. Name that name it like with a date and _01 or raw. Then along the way, crazy things can happen. Just save it as new iterations, number it sequentially, okay? So I'm going to number this like skull 01. And I'll save it as a Photoshop file for now. Now we're in a vector-based program. We'll make it smaller so we can see it. And I'm only going to do a couple simple things that I want you to, if you're working on a program, you follow along with me, okay? So what I want to do now is I want to recreate that scale bar and that figure legend so it's really clean and nice. You can do that in Photoshop as well. So for example, here's a line tool. And I'm going to pick there, line tool. And I'm going to make that color maybe this. That's what I want the line tool, or black, whatever you want it to be. And I'm going to hit here. I'll press shift so it goes in like a 90-degree angle or 180-degree angle. And what I'll want to do is match that perfectly. So, oops. So that's this right here. So this line segment, there we go. This line segment should match your original line. So you make it as long as you need to. And there, now it's a nice clean line, right? Text tool, sure enough, you can do that too. And I'm going to say, what is it? Instead of 1 inch, we want to go with, I mean, it'd be 2.54, but let's say 2.5 centimeters because we like to go metric in science. And maybe you don't like the color of that typeface, so you can change it to be black. Maybe you don't like the font itself, so you can change that. So you can change anything you want. Okay, and it's Big Caslin. I'm going to change that to, let's say, Calibri. It's here. Okay, and maybe I'll make it smaller. And I'll move it around so that could go there. So now I've created a couple of layers. Now on this layer, I still have that weird 1-inch pencil mark. I can erase that, I can paint over it, I can do it all kinds of ways with Photoshop. Illustrator, you just do something different, like make a box. So that's the idea, nice and clean. Now, figure legend typically would go here underneath. Maybe I want to move, oops, wrong layer. Maybe I want to move this and move that. Beauty of separate layers. And now I want to make a text box with a figure legend. And same idea. Figure 1, illustration of. And maybe you don't need to even say that. You just say anterior view of gopher skull. I hope I'm getting this right. That's what I can see. So let's say it's species. And sorry, but I can't read your name there. But what I'll do is I'll italicize this and get rid of that. Same idea. I can erase it. I can paint over it. I could put a white box over that as long as you choose the right layer. And I know it's that layer, right? So again, if I paint over or erase over, get rid of that. Yeah. So that's a simple dirty method in Photoshop. Another method would be, again, to take it into Illustrator, where if you want to be able to scale this up, it doesn't make sense if you don't, it's not as necessary if you have a raster-based image to begin with, like the skull. But say you created a vector-based image, then the ability to scale these up will be really, really important. Does that make sense?
How do you scale it for journal publishing formatting, I guess, or journal? Yeah.
So look at this specific journal and their image guidelines, and you'll find that they'll give you specific parameters, usually maximum length and width, or what it means to have a quarter page, half page, full page, something like that. And pay attention to those parameters very closely. And then as long as your image is produced at a larger scale than that, if it's raster-based, and it doesn't matter if it's vector-based, then what you do is you scale it as is appropriate given their guidelines. And different journals, for example, will ask for different formats. So some will ask you for the original, for example, Adobe Illustrator file versus a different format, sometimes just the flat out PDF. So the final version, what I do is I save things in multiple formats. Whatever I produce, I'll have the raw files as a Photoshop file, as an Illustrator file, and then I will export as a TIFF file without layers in the LZW, so it compresses well, but doesn't lose information. As a PNG file, PNG, and as a small JPEG file, so I can toss it around and send it easily. But ultimately, you don't want to lose a bit, an iota of resolution in the process. So TIFF is great, PNG is really good. But again, it depends on what file formats and parameters are required of the specific journal or publishing entity. Would people like for me to give you a display of different three-dimensional models and relevant books and things like that, and walk you through some resources as well as products that I've produced? That's option one. Option two, I play around digitally with more of your drawings. Option three, I go back to my original PowerPoint and pull up points that I skipped, and we go deeper into it. Or option four, open for questions, is just a Q&A. I may as well show you something really wild. So let's say I've got this drawing. Now, one fun thing you can do, you can transform it in all kinds of ways. You can scale up or down. You can rotate, skew, distort, perspective. And this is one that I've been playing around recently, Puppet Warp. Look at this. So with Puppet Warp, you can pin and change just little aspects of the illustration and warp it in different ways. Wild, right? So let's say I've done that. I did that. Now I'll transform in different ways. Like say, for example, now I want to skew it. And we haven't talked about perspective at all. But look at this. Now it looks almost like you're staring at the skull from an angle. And if I want to warp, this gives you a little bit more control. So edit, transform, distort. You can distort areas kind of infinitely in wild ways. So, oh, 3D models and resources. Let's do it. I'm going to close all this. Sharing some resources. Guys ready? I'll show you a handful of things. First, I'll show you models. So when I worked at, well, may as well start with helmets. So this is Honeycomb. Honeycomb built on a recycled bicycle helmet. And when I pulled that bicycle helmet out, I decided to make a paper mache mask at the bottom. So this was for Halloween, just a fun way to collaborate with insects. This is also paper mache up there. And then back in the days when I was working at the Natural History, what I do is I directly fabricate things. So say, for example, here's an HIV model. So human immunodeficiency virus. So HIV, we made them massive, right? This is just half a sphere. This was a test model I made. The others were obviously a full sphere. This was part of an exhibit called Epidemic. And in order to create this, I needed facts. So I wrote to faculty at Harvard and other places to find out what those surface proteins look like. And they sent me the information. Now you would just 3D print those probably, but what I wanted to do was directly fabricate one, because they're all the same. And then I took a styrofoam ball, coated it, I think it was plaster, sanded that down, and then I glued a billion of those features that I molded in silicone, cast in urethane, 4-minute urethane, glued all over. Now I wanted to make, that's great, I can make a bunch of those, but we wanted to make them glow, right? To glow and to have certain colors and be somewhat translucent. So what I did was I took my prototype, which was that styrofoam with the plaster, with these urethane blobbies, surface proteins on the top. And I made a 2-part silicone mold out of that, which means I placed this in something, could be clay, for example, and the top half, I poured a silicone mold over that business, pulled it, and I did the other half. So now I've got this 2-part silicone mold. I needed a mother mold or a jacket over that to hold it in place. Then it gets more complicated. I'll give you the short story, simplified story. There are several layers. So there's RNA represented by a green plastic in here. Then there's a translucent urethane that's a capsid or capsule that holds it. That was embedded inside this red icosahedral structure that I made translucent. That was embedded in this. So it was a multi-step process. But to make it simple, let's pretend that it was just one ball. I put that in a pressure pot and I mixed urethane with a dye, the color we wanted, and I poured that in the mold inside a pressure pot, cranked up the pressure, closed it up so that all the bubbles that would otherwise form would be crunched into oblivion. So these are a couple of urethane models that were cast from silicone molds, rubbery silicone molds. Here's dung, heap of dung. And the idea here is that I made a dung beetle rolling along a dung ball away from that ephemeral, valuable resource that is a dung heap. Now, how did I do that? You can make it out of plaster and roll in some hay, but the plaster you'd want to pigment and paint. In this case, I think I used Sculpt epoxy or something, or urethane, can't remember exactly. Maybe it's plaster. So let's say I make plaster. A friend of mine, colleague, dropped a big blob of plaster from a ladder from the height of an elephant's anus to get his dung heap. The idea is you want to pigment it first so it's dark. You mix it with straw or hay that wouldn't be fully digested by the elephant. And then what I did was I painted it partly, made it glisten-y with, say, a matte medium or other medium. And then I drilled holes in it and made beetles, more beetles to try to take it apart. Now, how do you make those beetles? The same way I'd make one of these. Colleagues of mine went to Central African Republic, collected these specimens, brought them back, and then my goal was to disassemble them, mold the various features, So that could mean taking the legs and making a two-part silicone mold of the legs, making a silicone mold of the body with the head, making a plaster mold of the wings, assembling all of the casts. The hind legs here are made of hot melt glue with wire inserted, so I can bend the legs if I want to in position, so it can go in a diorama dynamically. And the antennae, a very fine wire, so I drill into the urethane body that I cast from the silicone molds and insert the wire and glue it. And then I paint it in different ways. I'd spray it with an airbrush to get a base coat or two. I would go in with oil paints to get the markings. And maybe that means creating insect damage, fake insect damage on a leaf that this katydid's mimicking, right? The wings here are just part of the urethane cast, but the wings here, like wings I'd make on a model of a butterfly or another insect, I actually wanted to get the three-dimensionality of the venation as well as making them translucent so you could see part of the body through it. And the way I did that was I took the wings, placed them on a bed of, and you can use different things like clay, paper pulp, and I poured plaster over that. And the plaster now is a beautiful fine mold of that wing. I drill holes at key spots along the veins around that wing, place the plaster mold within a vacuum plastic machine that heats up a sheet of plastic above your plaster mold and then sucks it down over that mold. The holes help pull for greater resolution or accuracy in the wing. And then I pull it out, cut the wings out, and then I paint the wings separately, add a little wire, drill into the urethane body, and assemble. So those are some of the models, example models. Okay, I'm going to show you a few books, and I think we'll be out of official time. So the books I'm going to share include this whole series. So The Visual Display of Quantitative Information by Edward Tufte is a really great resource for understanding and even quantifying, and this is something we'll do in a future workshop, I think the second one, is to think about how not to lie and to quantify the veracity of your visuals. Envisioning information, he just has a really good series that goes into visualizing quantitative information. And then you've got fun things like pop-up books, like this one, which has outrageous pop-ups. This can be a really valuable, exciting, engaging tool for visualizing different subjects, right? There's a book called Inside Out, National Geographic Society, and each page is full of some amazing visual that looks on the inside of things. So, for example, this just came out, and I only found out about it a week ago, The Insect Artist. So this particular artist in Oregon has gone through examples step by step of how she approaches illustrating different insects. An old classic by Charles Papp is the Scientific Illustration, Theory and Practice. You'll see it looks pretty dated on the inside, but the manual techniques are still valid. The Guild Handbook of Scientific Illustration, again, is your greatest resource if you're interested in looking at methodological approaches, the business of being a scientific illustrator, and reaching out for resources. Then there are lots and lots of others, like Art of Botanical Drawing. Here's another thing about scientific illustration, booklet on scientific illustration, preparing scientific illustrations. A lot of them are moderately useful. limited in their scope, but if you use the resources through the Guild of Natural Science Illustrators website, I think you'll find some of what you need, or you can reach out to me. Here's a fun, crazy one. This series has 3D glasses, so anaglyphic 3D. So you just take your glasses and look at this complexity, and guess what? It simplifies with red, green, or blue filters, so you see 1 layer, 2 layers, or the 3rd layer. Lots of independent individual illustrators. Here's a great one. Paintings by Bernard Durin. And each page here is just a marvelous 3-dimensional insect. And you see the shadows there as well. And other arthropods. All right. Our time's up, but I'm up for endless questions, answers, or if people have requests, we continue with tutorials if time allows. And for those who have to run, thank you so much for joining me. This has been so exciting, and I want to thank Daria and Joanna and all those folks at Johns Hopkins for making this possible, because we could have had just a handful of people, and that'd be great too, but being able to make it freely available on this scale was my desire, because I want to spread the joy.
We do have 2 Q&As that haven't been answered yet, so we could address those.
Please.
So one is, at what point do you know if you need to cite an image you used as a reference to draw based on, or to get textures or colors or such from?
So, sorry, is the question if you've used elements like textures in your illustration, at what point do you need to cite the sources of those?
I think so.
Yeah, I don't think there are any hard lines or hard and fast rules there. It's kind of like sampling for music professionally. When do you step over a line? And I'd argue if it's at all recognizable from a very specific copyrighted source, then the onus is on the illustrator to attain permissions.
Okay, another question that's very specific to something you showed. Would it have been easier to scan a dung, 3D print it, and start from that? Any preference for taking a more manual approach?
Yeah, this is largely personal preference. So for example, there are sculptors like Alan Drummond who will create highly realistic magnified models of arthropods. And he does a mix of manual and 3D printing. So 3D printing is really powerful. For some artists, it's a tool that can advance their art in satisfying ways. For other artists, they lose that personal, tactile sensation that is a critical part of their art process. So for me, the years I worked making natural history exhibits were kind of just on the edge of 3D printing being really accessible. And I'll have to say, I really loved making manually the direct fabrication and the molding and the casting. Colleagues of mine who are still there at the American Museum of Natural History certainly do a lot of direct fabrication, exquisite work. They'll also incorporate some 3D printing. So really personal choice, but also it can be context dependent or project dependent. If you have to make 1 billion honeybee bodies, 3D printing, if it's cost effective and captures the detail that you require, could be the way to go.
There was a question about the use of AI as a helping tool for illustrations.
The AI question. This is a difficult one to answer because AI is a tool that can be used for good or evil. And as I told some of my students in classes this past week, we all use AI in some form or another, whether artificial intelligence underlying it, right? So it's not that we go all or nothing unless you're going off the grid and decide to not do anything that involves software programs that harness AI. And AI can be used as a really valuable, powerful tool in different ways. And I like the way that question was worded because it involves as an aid, as a tool, not a de novo and in toto production. Use it to guide me. In writing, you can certainly use it to guide you. Like, what way could I outline this paper? Or how could this be written from an AI standpoint? Does it work? Is it a good starting point, not a good ending point? Similarly, I think visually, people either deplore AI or harness it in ways that are pragmatic or practical or inspirational. So I'm not going to toss it out entirely, but to say that if you're working on a project that is either visually very complicated or you just want to get inspirational forays through iteration after iteration that's pulled through AI, yeah, maybe it could be a really great starting point or a guideline in some ways. I know that's vague, but when we think about AI as a visual tool, that's a huge umbrella. So it may be more pragmatic to think about case studies.
I think this one is interesting. Somebody's asking, how do you get your career off the ground for your average wannabe artist?
Artist or scientific illustrator?
This
It's
specific person says average wannabe artist.
okay. Well, we've got artists like my mom, I see her right there, who are professional artists. And I'm not a professional artist, right? I'm hired professionally as a biology faculty member, as an entomologist and animal behavior biologist at a university. Now, I really love to look at the intersection of science and art, and I love to produce art on my own. So that's taken me in really interesting directions. I guess you could say I was a professional artist or artisan when I was working at museum exhibitry or as a scientific illustrator or a model maker or this and that. Getting your start depends on what your goals are. So if you want to be a professional fine artist who exhibits in New York City, it's a very different thing than if you want to educate people with illustrative tools, either two-dimensional or three-dimensional works in museums or in books or in other ways. So that, I think we'd have to explore very specifically what professional directions excite you. For me, I knew I wanted to combine science and art in somewhat of an applied or educational way. And so I really targeted natural history display making studios, museums, and things like that. And I did some freelance work as an illustrator and a little filmmaking, and that was really valuable for me.
So this is actually a good segue because somebody's asking, how do you get into the natural science exhibit work? Like, how does one go about doing that? Where do you start, I guess, if you're already trained as an illustrator, for example?
Right. So sadly, the profession of scientific illustration, while much needed, vanishes in terms of professional positions at museums. So when those jobs are gone, like someone dies or retires, they're not being replaced. Now, you can still work at different firms, and I highly recommend you join Danielle Van Brabant's workshop on medical animation because that's her career. She is an artist. She's a medical illustrator and animator full-time. And so she can talk about how she built her career. In terms of working at a museum in exhibitry, yeah, there are some, like still at the American Museum of Natural History, at the Museum Public Museum in Milwaukee and Field Museum, there's still exhibit departments. And there are ways to get in that way, or through the Smithsonian, but freelance may be a way to go to start your own business. And there are many successful examples of those who work as independent illustrators.
We have a few more questions, but I think people are definitely leaving. So I think we should wrap it up for today, and we can save those questions, and we can, if you want, you can take a look at them later and address them.
Yeah, that'd be great.
Later time, yeah. So sorry if we haven't gotten to your question, we'll try to go through all the chat and pull out all these questions, and then we'll try to get all of them answered for you, all right? So
thanks, everybody. I hope this was useful and fun.
Yeah, I don't know if you can see the chat, but there are hundreds of messages coming in with thanks and compliments to your work and to you and the workshop. So
Thanks again, everybody.
all right.